Hello everybody, it's Miss Johnson. We're going to move on from carbohydrates. We're going directly into lipids in this video. So let's just lay some very basic foundation about lipids. It's not mentioned here on this slide, but I like to point out that they're made of the same three atoms that you find in a carbohydrate. So lipids are made out of carbon, hydrogen, and oxygen. And if that's the case, why then are they not carbohydrates? They both have the same makeup. And the reason is because of how the molecules attach to one another. So when we talk about emergent properties as far as like atoms go, this is a great example of how two things that could have the same atoms present do very different jobs. So we talked about carbs being a quick energy release. These are different. They're going to be used primarily for long-term storage when you're talking about just the fat part of lipids. But lipids serve some other key roles. So besides that, we also need to keep in mind that there's no like actual polymer name for a lipid. There are three types of lipids that would like fall within the realm of the lipid umbrella, I guess. So we'll talk about the three of those. What else? Oh, so all of them are hydrophobic, meaning they are water fearing, or they do not have like a love of water or a need for water. And I'm just going to break down that word really quickly. Hydrophobic. So hydro is water, and then a phobia is a fear or to be scared of, or like to not want something. So hydrophobic literally means that lipids do not appreciate water. They don't need them. They don't have to come in contact with them. So we find a repulsion of water from lipids. And so if we look at their structure, the reason why they are hydrophobic is because they have these really long hydrocarbon chains. And reaching back to the knowledge that you got from the earlier chapter or chapters, we know that hydrocarbon chains are carbons bound to hydrogens, and we know that the valence of carbon is 4. If the carbon is surrounded by hydrogen atoms, they're covalently bonded. So breaking that bond is tough. Like it would take energy to do it. And so for that reason, if H2O were to come in contact with a lipid molecule, you wouldn't see any dissociation occurring because the carbon is happy, the hydrogens are happy. There's no need for it to mix with H2O in solution. So that is something that they all have in common. Also, the bonds that we find in between the hydrocarbon chains are nonpolar. So, I mean, that's basically what I was just saying. Covalent bonds, they're nonpolar because the carbons are happy, their valence shells are full, and hydrogens are happy and their valence shells are full. So let's talk about the three different groups of lipids that will fall under the lipid umbrella. Those are fats, phospholipids, and steroids. Okay, so very briefly, let's just address fats. So this is the fat that you would see listed on the back of a nutritional label. This is the fat that you cook with. This is the fat that you actually consume. So these fats, they're made of two parts. They have a glycerol molecule, and if you remember anything from carbohydrates, we did talk about how things that end in OL, so they're hydroxyl groups, and they're made out of carbon, hydrogen, and oxygen, those are carbs. Glycerol is a 3-carbon alcohol, has a hydroxyl group attached to every single one of the carbons. And so what we find is that we can utilize the hydroxyl groups within the glycerol molecule and make them undergo dehydration synthesis with these things called fatty acid chains. So the fatty acid chain is literally, the front part of it is a carboxyl group, and then it's attached to a long hydrocarbon chain. So a lot of carbons and hydrogens, like a tail behind this carboxyl group. The carboxyl group and the alcohol from the glycerol undergo dehydration synthesis to create something called a triglyceride. In this case, it would have to be 3 fatty acid chains and 1 molecule of glycerol to make this fat.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long nonpolar hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules. The fatty acid tails in a fat can vary in length. A very important characteristic of fats is the number of double bonds in the hydrocarbon tails. Fats that contain only single bonds are called saturated fats. Fats containing double bonds in 1 or more tails are called unsaturated fats. Unsaturated fats tend to be liquid at room temperature. Vegetable oils are examples. Saturated fats, like butter and lard, are solid at room temperature. Saturated fats in the diet can lead to heart disease, while unsaturated fats are safer.
Okay, so that animation should have been helpful. The talk that the guy gave will lead us into some other slides as we go along, but he said a lot in a short time. So here's a picture of a glycerol molecule. You can see it. It's drawn here. All right, you'll notice that the glycerol molecule has the hydroxyl groups that they spoke about or that I just spoke about in the slide before. You see they're right here. There are 3 of them. And so here is that carboxyl group on the fatty acid chain there. So that's where the bonding is happening. It's happening between the hydroxyl group of the glycerol and then the carboxyl group of the fatty acid. They call this one palmitic acid. How do you know that it's an acid? Well, the fact that it has that electronegative oxygen up at the top, and we are essentially pushing out hydrogens when we break these apart. So this is just one of 3 dehydration reactions that would have to happen if we're going to make a triglyceride. So here it is when they're all 3 linked. Notice that the chain lengths vary, just like that was said in the animation. Another thing to point out is what it's called when you link the glycerol and the fatty acid chain together. It's called an ester linkage. You can still see remnants of the carboxyl group, and you do understand that the hydrogen that was lost from the glycerol here is in solution, which contributed to the whole acid portion of this. What else do I want to say? Really nothing about this one. So this is something that would be used for long-term energy storage. Like, it would stay. If you have ever gained weight or try to lose weight, this is what you're up against. Like, you have to be able to break these guys apart in order to I guess, like, decrease the size of a fat cell. All right, here's the whole thing, like, drawn out all together. So A is just the slide that was 2 slides back, and then this B part is the one that we looked at just in the last slide. All right, so what are they saying here? Fats separate from water because water molecules hydrogen bond to each other and they exclude the fat molecules. So they make it sound like here that just the water doesn't want to play nice, but it's not the case. The fat has something to do with it as well. Those, again, hydrocarbon chains are nonpolar. There's covalent bonding happening between the carbons and the hydrogens, and so you just get this oil or butter that doesn't want to mix with water. So we already talked about what a triglyceride is. It's literally just the glycerol plus the 3 fatty acid chains, and we can keep moving. All right. In the animation, saturated fatty acids versus unsaturated fatty acids were discussed. What you need to remember is that the saturated fatty acid chains, they are full of hydrogens. And so that's the way that I always remember it in my mind. I go like, oh, if it's saturated, it has a lot of something. And so in this case, it has a lot of hydrogen molecules bonded to the carbons. And then in an unsaturated fatty acids, because there is a double bond present, it may be that you are lacking only one hydrogen difference from the saturated fatty acid, but that one hydrogen molecule difference causes the atoms to have a property that is completely unlike the other. So what we find is that in an unsaturated fatty acid with a double bond, you get this kind of like kink in the fatty acid chain. And the best way I can describe it is if you think about like the bend of your leg, or like how your arm can bend. That's the way that that fatty acid chain would look if we were talking about an unsaturated fatty acid. Saturated fatty acids, if you just stick your arms out straight and don't give them any bend, what you'll notice is that it's really easy to just like stack things on top of it, and so you can get like this solid formation. Whereas with the unsaturated fatty acids, you get this like liquidy kind of a substance at room temperature. Okay, so this is what I just said in the last slide, that fats made from saturated fatty acids, they're called saturated because they are full of hydrogens. There's no double bonding, so you don't get that kink in the hydrocarbon chain. They're solid at room temperature. So examples would be like butter, lard, which is animal fat, any kind of grease or something that you use that is solid at room temperature. But plant fats, so like olive oil, canola oil, vegetable oil, sunflower oil, we can keep going on. And also fish oils they're liquid at room temperature. They're unsaturated fatty acids. All right, here's a structural image of what I was just saying with the arms being extended straight. So if you look at this particular molecule, because of the straight chains, they're able to stack on top of each other with like no problem. So you know that when something is cooling or losing heat, there's less motion in the molecules. So room temperature is still pretty good like movement going on, but in this case, at room temperature, this thing is going to become solidified, and it's not solid like you can't put your hand through it kind of solid. It's solid like you could scoop it and it would still be like a mass in your hand. It wouldn't like run through your fingers. So I know that that was like a point of contention, like, I don't know, a couple years ago when I was talking to a class about this. So I just want to make the distinction that when I say solid, I don't mean like solid, like brick hard solid. I just mean you can physically pick it up and it doesn't run through your fingers. So in the unsaturated fat, you'll notice that there are 2 fatty acid chains above the one that has a double bond. They're just straight fatty acid chains. There's no double bond in them. However, that very last fatty acid in the picture does have a double bond, and the double bond is right there, and that causes this kink that I was talking about, like an elbow bend or a leg bend. And what happens is when you have a molecule where you have this bend, the other molecules around it that have the same exact structure have a hard time sitting on top of each other at room temperature. They still kind of like float past each other a little bit. So you do have some thickness, because like if you touched olive oil and then maybe you touch water, they feel different. Like the olive oil is greasy, the water is not. So what is the point here? The point is that this kink in that last fatty acid is making it so that these molecules are just like liquid at room temperature. There's nothing else to really say. They can't stack on top of each other like that butter. And so this is what you get. All right, here's a picture of both of them on the same slide just for comparison. And again, these guys could stack on top of each other very easily. But this, with the kink, if I had one going this way, and let's say another molecule has it going like this, and then maybe there's one that's like that, they're not going to stack. They can't stack at all. They have to like move past each other. All right, and then this is just the callback to something I said at the very beginning, that the function of a fat is energy storage. Like this is literally the fat that would build up a fat cell. And I don't know, this is an interesting fact. You may already know this, but I'll just say it. So you actually don't gain fat cells as you gain weight. It's just that the fat cells become larger during like weight gain. And I'm not talking about like muscle mass gain. I mean specifically like an abundance of fat. So what we see is that because fat can be compact, especially when it comes from an animal, it's easier for your body to kind of carry that around. So conceptually, like it just works. Like it makes sense that lard would be solid at room temperature. And inside of your body is not room temperature. It is a little bit warmer. And so I'm not saying that all of the fat in your fat cells is completely solid, but the majority of it is solid. And yeah, that's that part. All right, so now we're going to move away from the fat that we eat and is on our food labels. We're going to talk about another type of lipid called a phospholipid. All right, so phospholipids have a phosphate group, so that should be pretty simple, right? Especially if you learn your functional groups, you know that there's a phosphorus atom and it's surrounded by oxygens. So in the case of a phospholipid, we're talking about that exact functional group being attached to a lipid structure. So they give you a definition in the first bullet point. In a phospholipid, 2 fatty acids and a phosphate group are attached to glycerol. So we're still working with the same hydroxyl atom / carbohydrate that we were looking at before. So there's 2 fatty acid tails. Again, they are water hating or like water fearing. They're hydrophobic. But because of the phosphate group attached, phosphate groups are polar and they don't mind coming into contact with water. So this is actually the type of lipid that makes up your cell membranes, and it should make a lot of sense when you see the picture. Okay, so this is the structure of the phospholipid. There's that phosphate group at the top that I was talking about right there. There's the glycerol portion of it that I read from the slide, and then here are your hydrophobic, the tails, the carbon, hydrocarbon tails. All right, and then they show you, so this is like a blown up version. If we zoom out a little bit, this is the kind of thing that they want you to visualize or see. So there's our model for a phospholipid. And then here they show you something called a phospholipid bilayer. So these are just close-ups. I want to go ahead to the phospholipid bilayer. Okay, so here we are. This here, so all of those purple heads that you see, those are all phosphate groups, or the portion of the phospholipid that is polar and that is not water fearing. So these are actually called hydrophilic. So they like to be bathed in water, or they can withstand being bathed in water. And what's interesting is the hydrophobic part, they are like they're all stacked together like in the center. It kind of looks like an egg with legs to me. So I'm gonna say that the egg head part is okay with being surrounded in an aqueous solution, but the feet part, they don't really want to get wet. So what does this make up? It makes up your cell membranes. And why would you want that to be a cell membrane? Well, your body is made of water, so you have a percentage of you that is water-based. So what this means for your cells is that water is not going to just easily slip in and out of the cell. There has to be some kind of like channel or port for it to go through, because the hydrophobic tails are going to repel the water. So your cell can be bathed in water. You know that inside of your cells there is some like a water-based solution. However, you're not losing the contents within the cell because it can't break through that hydrophobic tail region of the membrane. And likewise, water cannot just flood into the cell because of those hydrophobic feet that are in the center of the membrane. So it creates a nice boundary between, they say this eloquently. The phospholipid bilayer forms a boundary between a cell and its external environment, which is exactly what you want. All right, let's go to the next class. So we did fats. We know fats, we eat them, we can store them, we use them for long-term energy. We know that the phospholipids are used to make up cell membranes. We know that there's a phosphate group, and then there's some feet that are hydrophobic, the fatty acid chains that will not allow water to just freely cross the membrane. Then you have the last category here, so steroids. People think about steroids and they think about like anabolic steroids, like people who I don't know, like they're bodybuilders and they take steroids. Or maybe you think about steroids when you think about, I know when people get like bronchial infections, so like, you know, something wrong with your lungs, they can give you a steroid which should help like with inflammation. There's also some other steroids that you may be familiar with at this point just because of some things we've done in class. So like estradiol and testosterone, those are two steroids as well. But we think about them more as hormones. Okay, why are we calling them steroids? The reason has to deal with strictly their chemical structure. So chemically speaking, a steroid is going to be a lipid that has 4 fused carbon rings within the structure. And the main one that we look to is cholesterol. That is like the mother of all steroids. Just like glucose is like the mother of all carbohydrates. So steroids with their fused rings, they can make cholesterol. Okay, I said that. Okay, and then what happens is you need cholesterol. Like, it's not like you should have a 0 cholesterol count. There is a distinction between like good cholesterol and bad cholesterol. I'm not going to get into that because we don't need to. Let's just talk about it chemically. So in this case, if you do consume too much cholesterol, it can like cause a clogged artery, and they call that atherosclerosis. It's here in the bottom bullet point, bam. So okay. It's essential in animals, but high levels in the blood can contribute to this clogging of your arteries. And a clogged artery equals like a blockage. And if you get a blockage, then the circulation is thrown off. And so that's why you have people who can die essentially from their cholesterol levels being too high. All right, so here we're going to look at an animation. This is just a 3D visual model of what cholesterol looks like. This particular model is drawn with all of the atoms, like it's a space filling model basically. The next one though is just the skeleton of what the molecule looks like. So here it is, same exact molecule. The only difference is it is not space filling, and what you should notice as it moves around is you see those carbon rings in the center, but you have like these pieces that branch off of the 4 rings, and that's what's going to give us variation within the realm of steroids. So this is why everything that is a steroid is not cholesterol. So here are the 4 rings. You can see them. They're really easy. 1, 2, 3, 4. All right, but what is important here is that all the other steroids we talk about are going to have the same 4 rings. Their differences will come based on what functional groups are attached to the 4 rings. So in this case, you guys see like a hydroxyl group is here. You have a methyl group right there. You've got a couple of other methyl groups. You know that this long chain area, so I'm going to highlight the region all in here. Remember that each point on this particular diagram, not this one, but like all that do this, there's a carbon here. It's inferred that there's a carbon here, and it has the correct number of hydrogens attached to it. So in this case, it would be 1 up and then 1 down. And that would be the four bonds that it would need to form to be happy. So that's it. That's lipids.